talk about. So I'm going to talk about recognizing risks and mitigating risks from giving gene therapy, and just help it give just a basis for understanding the physiology of that. And this will really center on sort of known risks, and I think Dr. Finkel after me will cover some of the other things. So we start off by just really understanding what these therapies are, right? And so you see a wild type AAV here with its wild type genome, and you see an AAV gene therapy here with its transgene genome. But these are viruses, and there's inherent risks that are not insignificant to these therapies. Those risks are based both on the innate and adaptive immune response to the therapy, and that can include an immune response to the capsid or an immune response to the transgene protein. This is, and we've already sort of seen this, but the FDA-approved gene therapy for SMA on SMN parvovec. So that's an AAV9 capsid with a human SMN1 transgene and the promoter. So you have to think about these things. The promoter is an important promoter. It's a ubiquitous promoter, so it'll be producing this gene product everywhere that it integrates, or everywhere that it's transduced. The dose is 1.1 times 10 to the 14 vector genomes per kilo. Here's the DMD product that's available, delandistrogene moxeparvovec. AAV, so it's AAV RH74 capsid, so different capsid, a microdystrophin transgene on an MHCK7 promoter, so a skeletal muscle promoter. So even if this successfully transduces other tissues, it's going to really only be expressed in skeletal and cardiac muscle. Again, the dose here is 1.3 times 10 to the 14 vector genomes per kilogram. And I just put in this little box to try to help you think about how many things is that. It's like 1,000 trillion things. So take 100, so it's 133 and 12 zeros for the DMD product. And the only number I could think of else that's really high is the national debt. So this is the national debt. It's tenfold less than what we deliver for a viral gene therapy. And remember, this is a therapy we're delivering based on a dosing vector genome. So we're actually dosing the number of genome, and you can have empty capsids there. So as you think about toxicities, think about that therapy all by itself. Consider the dose, the empty capsid number. There's a lot of manufacturing issues that go into this, and this is why it was important when she talked about the studies that were done with one manufacturing process and then now using a separate manufacturing process. That may be more trivial when we think about small molecule therapies, but for gene therapy, it's really critical because there's impurities there. You don't always package a full genome in every capsid, but yet we're delivering a dose based on just the vector genomes. But it's not just the vector itself. There's host factors that are important in this too. So the general health of the patient at the time they're infused, their age, their genetics. But we do see some predictable timelines, and these cross over any AAV therapies. And so week 1 is mostly an innate immune response to the capsid proteins, and then week 4 to 6 we see some other responses, and we'll go through these in a little bit of detail. Those are more represent a shift to an adaptive immune response. To mitigate risk, we treat with prednisone. So we take the bluntest hammer we can find and we just hammer away at the immune system with that to try to sort of calm the immune response down. And we do really close laboratory monitoring. It's important to know that clinical symptoms are always not always present, so that laboratory monitoring is really important as we sort of anticipate the possibility of adverse effects. There are some research protocols that use other immune suppressive therapies like rituximab or sirolimus or other immunosuppressives, but those aren't widely used in clinical situations right now. And then once you have an adverse event or a serious adverse event, there's very little data to suggest how do you manage those adverse events. So, we just increase the oral prednisone, that's easy. We bring you in the hospital to give you IV methylprednisolone, and after that, it's anything goes. And we've tried lots of different things at our center. I've listed a few here, but anything pretty much goes. This is a figure from Barry Burns' lab at Florida in a paper they just recently published talking about thrombotic microangiopathy. But I think you should go find this paper and read it and get to know this figure intimately, because this really encapsulates what happens after you infuse. So it's, number 1 there is infusion of the vector, and then what is the immune response as it leads up to antibody production, an IgG against that viral vector. And there's a lot going on. And the reason I put up here, because we don't have time to really go in depth, but it's a really important figure. And this, if you go back to it, you'll, I think, begin to understand the complexity of what's happening when we infuse these therapies. There are a couple other papers I just wanted to point people to talking about serious adverse events in onasemnine giamma therapy for SMA. These are from real-world data on the Restore registry here, but they all kind of fall into some of these more common things that we often see. So if you look just here, we see hepatotoxicity, transient thrombocytopenia, cardiac adverse events, and thrombotic microangiopathy. This is another paper out just late last year from the UK with similar set of real-world evidence in these type of adverse events. So we'll just kind of go through some of those quickly, and we really don't have time to do them all justice, but just to give you an overview of what's possible. And I do think you need to know what's possible before you get started in these things. But nausea and vomiting is common. It's day 3 to day 5. It's seen in most every patient. But just remember, nausea and vomiting can be a symptom of heart failure. So when we think about these things, it's pretty simple to treat dehydration, but this may be a harbinger of other things. Myocardial injury being the most important of those. And I do want to just make a point that probably we move away from the terminology of myocarditis. As we've seen some of these cases come up, they tend not to be particularly inflammatory, but they are myocardial injury, and you see this troponin elevation, which is really common in almost every patient we treat with AAV. But the thing that can happen is that can really get out of control and get rapidly progressive cardiac failure leading to death, and we've seen that in 2 different cases now, and I'll give you a little more detail on those in a minute. Thrombotic microangiopathy, also a relatively common thing that we've seen in certain of the gene therapies clinically. That's low urine output and evidence on laboratories of hemolysis and thrombocytopenia, and that can lead to renal failure, especially if it's not recognized early. That's largely a complement-driven phenomenon. Just to kind of review that sort of space in your head. So in most cases, this is around the end of the first week post-gene transfer. We see a drop in the hematocrit, drop in the platelet levels, and ultimately a drop in renal function, and we can see renal failure. This has been seen in multiple different AAV platforms, and the risk of that may be somewhat dependent on the nature of the clearance of those AAVs. I think we're just starting to get some understanding of that. And that's a complement-mediated reaction to the capsid proteins themselves. And that we've now, I think, in the community developed some experience with treating this with early treatment with eculizumab. I think we do better. But I think, again, we have a lot to learn. Acute liver injury is probably the most common thing we see in this 4 to 6 week time period. If you're looking at the DMD data, and we don't have a lot of this, but from the FDA briefing document, about 37% of cases had a bump in sort of a transaminitis. 5 to 7% of those were hospitalized. This is from probably a mix of the Sarepta studies. But it's not that rare, I guess, is the take home there. It's often asymptomatic, so that laboratory surveillance is really important. And as you think about the timing, you've survived the first and second week, and so sometimes you might be thinking you can take a deep breath, but this is going to hit hard like 4, 5, 6 weeks in. So you have to be really vigilant about that. We follow AST and ALT. They're even helpful in Duchenne patients where we know they have AST, ALT elevations due to their muscular dystrophy, but they get a step way beyond that, right? So if you know those baseline numbers and following them, it's okay. The GGT also responds, but it's slower to onset and slower to recover in our experience. We also follow bilirubin levels, PT, PTTs for synthetic liver failure as well, which can happen. These are usually responses to high-dose corticosteroids. In our hands, for someone who doesn't respond to an initial bump of from 1 per kilo to 2 per kilo of oral prednisone, we bring them in the hospital. Certainly no less than 10 milligrams per kilogram per day, and we've treated as high as 30 milligrams per kilogram per day for 5 days in some of the patients. There are at least 2 deaths that are known, and not many details known about these patients, but attributed to liver failure in SMA patients. There's 4 deaths that we heard about in the myotubular myopathy trials. And there's probably lower risk in younger patients. This is the sort of paradigm that we've been working with at our center, that we felt pretty comfortable with these newborn babies that we're treating. But then in the fall of 22, we had 2 babies in a row, both treated under 21 days, both had the same sort of pattern of a marked elevation of liver function in AST/ALT at that 4 to 5 week kind of interval. So it can happen really in any patient. Just another word about, about immune myositis. So this is now represents something of a shift in what the immune response is. Everything up to now has been really an immune response to the vector, to that capsid proteins. And immune-mediated myositis, this comes up around weeks 4 to 6. There's a really nice paper published by Dr. Bonneman's and a group of multiple sponsors who all experienced this around the same time in their DMD studies. So 5 patients were observed. They all followed this similar pattern. They're dosed with different vectors, different doses, but all in that 3 to 6 week period post-infusion had a severe sudden onset of proximal muscle weakness leading to loss of ambulation. And in some cases, bulbar weakness that led to respiratory insufficiency and assisted ventilation. In 3 cases, there was some myocarditis involved with that. In all cases, they resolved, at least 3 cases, 2 were resolved with sequelae, and treated with a variety of different immunosuppressive regimens. And these are really thought to be an immune response to the transgene itself. And so when this group of people brought together by Dr. Bonham and Dr. Montoni got together, what they saw was this, and you'll see this figure in their paper. On the bottom is the mutations in the patients who had this adverse event, all had large deletions that all overlapped and included the C-terminal end of the gene in an area where all of the different microdystrophin constructs had the same protein. And so what they've done some additional work to show that these are really a T-cell mediated response to the transgene. So you turn on this transgene, you have no immunotolerance for this novel DMD protein, and you make an immune response to this. So this will be really important, and this is the thing we aren't likely to see in SMA, since patients with SMA all make SMN1 protein. So this can be really disease-specific sort of response. So I just wanted to cover these 2 deaths in the DMD patients. We really don't have time to get into all the important details, but just to give you a thumbnail, this first one was on a Pfizer trial, 16 years old in a non-ambulatory part of the trial. He was treated with 2e14 vector genomes per kilogram. At that point was the oldest, but not the heaviest kid to be treated. He had baseline cardiac MRI findings that did not meet exclusion criteria, but did note wall motion abnormalities and gadolinium enhancement, which I think is an important piece of the puzzle. He had his first troponin elevation that was first seen at day 2, but we didn't actually have that result until day 5. And then follow-up troponin testing showed marked elevation in his cardiac troponins. We saw him for rapid cardiac evaluation after that. He looked really terrible, and through the course of the following day, he just had sudden cardiac complete failure and died hours later. We don't have autopsy death from that case, but we do have autopsy case from this other case, 27-year-old non-ambulatory Duchenne patient with a really N of 1 therapy. So this is a therapy tied exactly to his mutation in DMD. Again, met screening criteria for his cardiac MRI of greater than 55%, but did note wall motion abnormalities and gadolinium enhancement. He was pretreated with steroid and sirolimus, got a similar dose to what we see in the other therapy protocols. On day 2, had a downward trend in platelets, and that triggered a series of eculizumab and other immune suppressive therapies, and then trending up BNP and troponin levels over the next several days, which led to a drop in his left ventricular ejection fraction to 45%. He started on BiPAP on day 5, and despite a series of interventions, immunotherapeutic interventions, he had continued worsening of his cardiac function and developed a pericardial effusion, and then died shortly after, or had cardiac arrest shortly after coming off his BiPAP, very briefly just to go to the bathroom. Autopsy findings there were interesting in that they didn't really show inflammation in his heart, so that just mass infiltration of inflammatory cells. They did see in the lungs evidence for Hyland-Cast, so ARDS type of picture in the lungs. And likely what happened is that this patient had just third spacing and once he came off of that BiPAP, the lungs collapsed and then he went into cardiac arrest in a very vulnerable heart. So these things can happen. They can happen very rapidly, and you have to be on it. I did just make a quick summary of the myotubular cases, but you've heard that already. I think the interesting part of this, though, is that it's a very severe disease that had a very good response early in the study. And as the study progressed, and you heard about those deaths that had happened, that first in 2 patients and then a third, and then when they restarted again in a fourth at a lower dose. But what really that led to was a really careful evaluation of why did this happen in these patients. And what was seen is that this therapy really unmasked a latent hepatobiliary disease in many of these patients that we really just didn't appreciate previously. So there's always some sort of surprise at the end of the day with these therapies. And as we get deeper into gene therapy, we just need to be ready to address those things. And so to me, there's some lessons learned from all this. One is that gene therapy is safe like chemotherapy. It's safe in the right patient at the right time with the right people following, and we need to be very careful how we do these things. And we get these really excellent results, but the potential for bad things is really high, so we just need to be very careful as we do these things. But these known events, they can often occur at a predictable timetable, so we can be attentive and vigilant in following them. So the early close surveillance is really important. There's likely some, other factors that we just don't know about. So doing deeper investigations into what patient factors might lead a person to be more susceptible to these things are really important. I think one thing we learned from the 2 patient deaths is that a left ventricular ejection fraction as an exclusion criteria is insufficient in monitoring safety for these therapies. And we need to think about really their underlying cardiac physiology and what is the underlying health of their heart. And there's no really good, clean way to do that. But we need to know those wall motion abnormalities, that gadolinium enhancement, what amount of muscle mass that's left in the heart and things like that before we really push more deep into those therapies. And we really have a need to develop best practices around how do we manage these adverse events. And just have a slight dose of humility, because when we see the SMA pictures, when we see these DMD pictures, that feels really good. And we know we're doing something good, but we just need to just know that these things can cut both ways. So I'll end there, and we'll go to Dr. Finkel.
Neurologist. I focus on children with neuromuscular disease, and I work at the St. Jude Children's Research Hospital in Memphis. And my assigned topic was potential short and long-term harms of gene therapy. So this is going to be more of a little bit theoretical talk with some data thrown in to try to highlight some of the emerging points that I think will be important for us to keep in mind as we're treating patients with these gene therapies. And I'm going to limit the discussion to AAV-mediated gene transfer therapy, not to say that ASOs and small molecule drugs, which are gene-targeted treatments, are not relevant, but for today's discussion, we'll limit it to AAV. So, as you've heard already from the prior 2 talks, with gene therapy, we're struggling to find equipoise. We're trying to find that right balance between safety and risk as we're identifying it, and efficacy. So when we look at the preclinical data, it's helpful but not entirely predictive of the risk profile in humans. So yes, we learn more proof of concept that the gene therapy works, from the fly all the way up through the worm, rodents, rabbits, dogs, sheep. Non-human primates are particularly informative, but none of them have been really fully predictive of the response in humans. And I'm going to give you some examples where what's been identified in non-human primates raised a red flag, and then how do we address that is really the question when we're treating our human patients with these viral vectors. Now, the FDA has certain safety requirements, of course. So we know in all drugs that are in clinical development to look for malignancy and cardiac and neuropsychiatric and other issues. But there are particular requirements, I'll just remind you, for children. That's called Subpart D, because children cannot give informed consent themselves. The parents are the surrogate for the child. And now even the field is starting to move towards considering fetal therapy. Can you treat a fetus identified with SMA in utero? And that's an emerging topic, and there's a special part of the FDA guidance requirements called Subpart B. And that's where you're really treating two patients, of course, the mother and the fetus. So that has even special requirements. And you'll probably hear more about that as this topic emerges. Clinical trial limitations are kind of obvious. When we design a clinical trial, it's largely with a homogeneous cohort to try to really identify efficacy and then the safety in the context of that narrow slice of the population, but it may not be representative of the disease population at large. So take, for example, the gene therapy with the accelerated approval for Duchenne in 4 to 5-year-olds. Well, there's every indication that it should work in older and younger patients and even non-ambulant, but does it have the same safety profile? How well it work? So you really cannot extrapolate so readily. There are certain limitations. The other thing is that we try to thread the needle with clinical trials. We make them long enough to be able to show safety and efficacy, but short enough that we can get those results and then hopefully get regulatory approval so that we can provide it commercially to the broader population. So that really pushes the need for post-marketing or post-approval experience, and this is done two ways, either through registries or through formal phase 4 studies sponsored by the pharmaceutical company. And so far we're learning that this is really important, and I'll give you the example that in SMA with the onasemagene abepervavac, we didn't really know about TMA as an issue through the clinical trials. That emerged shortly after regulatory approval. And the drug company, Novartis, took that very seriously, and they explored that, and they published on it, and they shared that. So now it's common knowledge, as you heard from the prior speakers. We know to look for that, but it wasn't something originally identified. So under short-term safety, you're familiar with the phase 1 studies, and then there's the phase 2, proof of concept, and then there's the phase 3, the large study. The point to be made here is that the phase 1 studies are being designed to help with the application for regulatory approval. And again, going back to the onasemagene abepervavac, that was approved by the FDA largely on the basis of a phase 1 study in 12 patients because the results were so spectacular. And the safety profile was really good. But again, it was 12 patients. Now, there was a phase 3 study as well to support it. But we're moving quickly with trying to get these therapies for children that have genetic diseases with high unmet need. The other point is that we kind of skip over phase 2. We go from a phase 1 or a 2A proof of concept right to a phase 3. So it's a little different from a standard small molecule drug with phase 1, 2, 3. Okay. So what about identifying risk? Okay, I'm not going to belabor the Aspero study because you've heard about that from the two prior speakers. But I think the important thing is that there were underlying risk factors that weren't known about at the time. But when you see a side effect, is it attributable to the drug or is it progression of disease that was unmasked? Or in some cases, a combination of both. When you think about the risk factors, there's pre-existing antibodies that have to be looked for. And these could be neutralizing antibodies or not neutralizing, but still, each of these diseases has an exclusion if you have a pre-existing antibody to that viral capsid. But they differ. It could be 1 to 50 for one disease. It could be 1 to 400 for another disease. So, but that's important. And I think what's also been learned is if you don't make any of that protein at all, and this was from the Pompeii studies early on, what's called CRIM negative, you might develop, you might have a reaction to the transgene and to the product, the gene product, as was mentioned earlier. So there's the immune response both to the capsid and the transgene that need to be considered. And then I just showed here a different cartoon that shows the early rise in the innate immune response, typically within the first week. That's complement mediated. And then the acquired immune response, as you heard earlier, usually starting at around 4 to 6 weeks. That's an acquired immune response, T cell and maybe B cell as well. So when you think about the actual viral vector, what's being used to deliver your cDNA or your gene to the target tissue, there's several factors to be considered here. What is the dose? Now, intravenous, it's per kilo. Intrathecal, it's a fixed dose. It's so many viral capsids independent of your size or weight. So there's an important difference there. And as you heard earlier, the manufacturing process is tricky, and it's improving, but these are still not pure manufactured or filtered. So there's always some degree of empty or partial capsids as well as the full capsids. So the total amount of what you give is potentially immunogenic, but it's only the full capsids that are really going to be therapeutic. There are other topics that I'm not going to go into any detail, things called CpG motifs that can activate toll-like receptor 9 activation, and that can cause an inflammatory response. There are concerns with off-target reactions, and AAV is supposed to sit in the nucleus as a separate little episome, okay? And then it's just going to spit out the RNA that's going to go into the cytoplasm, and then it's going to generate your protein of interest. But there's always some concern that are these non-integrating or are they integrating? Like lentivirus is designed to be integrating. But here it's not supposed to be, but there's some experts who feel that there's even some risk here. The other important thing that we need to keep in mind is that when you're treating with AAV, you start the steroids typically the day before, and even in some studies you're starting other immune modulatory drugs even before the gene therapy is given. The REGENXBIO would be one example for Duchenne. And then they're on a pretty high dose for a month or two, and then you're tapering down. So there have been some deaths due to the patient being immune suppressed and getting a superinfection. So the death wasn't due directly to the AAV vector, but it was because they were immune suppressed from the steroids. So how you manage the patient is particularly important. Do you put them in a bubble or a lockdown situation for a couple months? That's not really so feasible. But you have older siblings coming home from school with their snotty noses, and there are real issues in how do you manage these patients after the treatment. The actual administration of the gene therapy is a piece of cake. It's done in an outpatient setting. They go home. It's monitoring them, as you heard from Dr. Butterfield, monitoring them very carefully, even without symptoms, and trying also to avoid them getting superinfected, which is not so easy. So what's different about viral vector-mediated gene transfer? Well, it's obvious once you give it, it's in there. You can't take it out. Now, there are some efforts being made to turn on or turn off the gene expression. We're not quite there yet, but that's coming. And there are also efforts to try to de-target certain tissues. And you'll hear in a minute about dorsal root ganglia toxicity issues and risk. And there are efforts being made by the Wilson Lab at Penn and others, Bev Davidson's group, to try to say, can we steer it away from the DRG and try to make sure it's going to, say, to motor neurons or to muscle, and try to avoid other cells that are particularly vulnerable. And then there's some common themes, and you've already heard about this. I'm not going to go into it again, the hepatotoxicity, thrombocytopenia, and TMA. The myositis that's not always myositis or myocarditis, they're not the same thing, as Dr. Butterfield mentioned. There are also some vascular factors here, and as we're learning through these registries, sometimes there are newly identified adverse events like necrotizing enterocolitis was reported in 2 babies treated with onisamidine. So we're not sure yet that we have the full picture, and we can't let our guard down is the bottom line. I want to turn now to some issues that have been brought up from the animal studies. And these are some general toxicology concerns identified in non-human primates. Okay, so this started, this is a paper that John Day and colleagues put together and it's a meta-analysis, but also looking at particularly at non-human primates and focusing on osamagine epiparvovec. And you've already heard that there are different labels for the FDA and the EMA. So there are different doses that are being administered potentially for patients in the US and in Europe, and there will be different risk factors. The payer policies differ as well within different countries, as you already heard about. But the safety profile is what I want to focus on here. So there's a maximum tolerable dose, MTD, that's been identified in mice, but not in non-human primates and not in humans. So we're not really sure how to translate that. So that the dose in mice was 1.5 e to the 14th vector genomes per kilo. Okay, but what does that really mean for humans? We're not sure yet, but we know we need to look for cardiac thrombi, which fortunately has not been seen in humans treated with osamagine. And then, as we've already went through, there's the early and the intermediate issues, and then there have been some deaths, and we're trying much less than 1%. So if you think, okay, yeah, we're doing all this work to monitor these patients and make sure we're giving the drug and following them responsibly, but there have been, out of about 3,000 patients that the last time I saw it reported, and there may have been 5 deaths, but not all due to the, as I mentioned earlier, due to the osamagine. Some were the superinfection. So you really have to look carefully at this. Okay, I want to talk now about DRG toxicity, because maybe you've heard about this, and it's still an evolving topic. Okay, so this came out of the Wilson lab in 2018. Okay, so they treated 3 non-human primates it's not identical to the osamagine, but it's similar. Okay, and of these 3 monkeys, one had liver failure, and there were dorsal root ganglia changes in 3, and then in 3 piglets, they all had DRG ganglionitis, as well as clinical features. Okay. And then after that, the same Wilson lab did a large meta-analysis of 256 non-human primates. Now, these non-human primates are little monkeys. They weigh around 2 kilos. Okay, they're little things. Okay, so think of them about the size of like a newborn baby or a young infant. There's some studies in older monkeys. They usually cynomolgus or rhesus monkeys, but they're usually small and juvenile. Okay, but in that context, there was DRG toxicity seen. Now, and this is the important point. With intrathecal administration, it was 83% of those monkeys, and with IV, it was 32%. Still substantial. But the second point is that it seemed to peak and then it waned. And in most of these cases, that it was not clinically evident in this particular. And it seemed to be independent of the capsid, the promoter, the transgene, the age, the sex, the dose. Okay, so that raised a lot of questions. Then comes along this paper by Olivia Pellizzoni's group at Columbia. And what they did was they looked in mice with a construct that was similar but not identical to osamagine because it had a different promoter. It had a GusB promoter instead of the chicken beta actin, the CBA, which is in the commercial product. And there's an argument going on whether the GusB is a stronger promoter. But nonetheless, what they showed was that at a dose that's higher than what typically would be given clinically in children, and it was given ICV in the mice on the day of P0, day of birth, they did show initial improvement in the very severe mice model, but the important thing is they saw a late decline at about 4 months. These mice started getting weaker, and they showed both dorsal root ganglia and motor neuron loss and inflammation. And in the cells, they saw aggregation of the SMN protein in the cytoplasm, and they were able to demonstrate also it dysregulated the RNA processing of the spliceosome, and there were all sorts of inflammatory changes. So this raised a bit of a red flag. And then the question is, well, is this relevant? Now, Arthur Burgess at The Ohio State University has done a parallel study, and he tells me, I guess personal communication, because it's not published yet, but he told me that he was not able to replicate this using the CBA promoter. So he thinks it's a promoter issue. It's not due to the capsid or the dose. But we'll wait to see how that shakes out. But in the meantime, I want to highlight a study quickly that was done by Novartis. And it's a very responsible study. And what they did was they took different groups, and I'm just going to highlight a couple things here. One is when they treated intrathecally, this is the important thing, intrathecally, they saw that neurofilament levels went up, and then they came back down. Okay. And then at about 13 weeks, when they looked at the tissue, there was marked inflammation and neuronal degeneration. And then if they waited and looked at another group of monkeys at 52 weeks, and this was in the cervical DRG, and that's with this red line. So at that point, the neurofilament levels had come down. They've normalized. But there's neuronal loss and there's some gliosis. So what does that really mean? Well, we're not sure. Now, this was also given at a higher dose than what we would be giving clinically. So it's not clear how relevant that is. But just to move forward, this is at an even higher dose. Okay, so this is like a megadose. But what they showed was kind of interesting. At 6 weeks after intrathecal administration, okay, of what is onisamogene epaparvovec, they saw all sorts of neuronal changes and inflammation, as you can see on this H&E. But look at the immunohistochemistry on the right, this one here. And you can see that the cells that take up more of the viral capsid, that's the brown staining, are the sicker cells. So what they showed, and their conclusion was that when you give a gene therapy intravenously, or in this case, intrathecally, it's not distributed, it's not taken up uniformly by all the cells. Okay. And some of the cells take up more and some take up less, and the ones that take up more seem to be more vulnerable to toxicity, which is what's shown here. So that's just something to keep in mind. And John Day, a colleague, has been making the case that we really need autopsy studies. So we never want to see our patients die, but particularly on patients who are treated, we need to see the distribution of the viral vector. We need to see the expression, the inflammatory gliosis. All of this needs to be looked at in patients treated with clinical drug. So I'm not going to talk about DMD. You've heard about that before. I'm not going to talk about the CRISPR. You've heard about that also. So I'm just going to wrap up and just say that despite these words of caution, AAV vectors for gene transfer can be highly effective, even when administered at a high dose. But there's a narrow therapeutic window between nailing it where you're getting efficacy and avoiding toxicity. And that the preclinical models are helpful, but just to a degree. The clinical trials are often limited. We need to have these post-marketing registries, et cetera. So that would be my plea here. So with that, I'm going to stop, and I thank you for your attention.